start to pick up with our work. So, as part of our routine barcoding, we have turned up all of these invasive or introduced species. We have Gracilaria vermiculophylla in British Columbia and Rhode Island, Masiella japonica in British Columbia, Genigongus griffithii in Australia, Eosiphonia japonica in Rhode Island and Australia, and on and on and on and on the list goes. Almost all these species come from Asia and get moved to other parts of the world for some reason. I haven't figured that part out yet. There are some exceptions. There's a Serranium here that's actually from Europe and has been introduced down into Rhode Island. The issue here is that these things can look like the plant that's supposed to be in the area. So this Gracilaria vermiculophylla has been completely overlooked in British Columbia. It looks exactly like the endemic Gracilaria pacifica that's there. You can't really tell them apart, even with anatomy. And when you get into these bays and estuaries where we find this thing, it's actually the dominant species. It's displaced completely the one that's supposed to be there, and nobody's even noticed because they look exactly alike. So it creates some interesting problems to consider. This one has me worried. This is Heterosiphonia japonica. 2007, 2009, we only found it in Connecticut and Rhode Island. 2010, we found it on the southern part of the Cape. 2011, we found it on the northern part of the Cape. And this year, we were diving in northern Massachusetts, and the entire bottom is coated with this thing. This thing is a real weed. And it's moving north, and it seems to be doing better the colder it gets. So I'd be keeping an eye out for that one in this area. But this is not a doom and gloom talk. You know, we find introductions and invasives and this and that. We also find some cool things that are just fun to think about. So one of the patterns we had that we couldn't explain is we had species that we found in California, and then we found them up here in Guayhanas in northern British Columbia, and nowhere in between. And this really had me scratching my head. I mean, were they taking a plane up there? How do you get species here and nowhere in between? And Norm Sloan, who works for Parks Canada up there, said, I found a red abalone shell on the beach up here in Guayhanas. Red abalone grows from southern Oregon south, or central Oregon south. So that abalone shell somehow made a 1,000-kilometer trip from central Oregon at best to Guayhanas. And the question was, how did it get there? Did it strap onto a boat or something? He said, there's a little bit of kelp holdfast on the shell. Can I send you this little bit of kelp holdfast, and can you sequence it for us and tell us what it is? I said, oh, absolutely, send it down. And we sequenced it, and it's a floater. It's an Nereocystis. These things float. So what happens is these little baby Nereocystis get on the abalone, and these things grow and grow and grow. Nereocystis can grow up to 2 meters a day. These plants become 30 or 40 meters long with this massive float on the top of them, and sooner or later, the little abalone goes, and away it goes. Whips up the coast on the current, and on this abalone we also get other types of seaweed. And so we think what we've got is this rafting of algae running up the coast here, and that was pretty cool to figure out. This is the first real data that there's rafting occurring as part of the distribution of algae along this coast. And it was a fun little study that we got to do because of our DNA tools. We can't identify a little bit of the kelp holdfast by morphology. They all look the same. Okay? So tomorrow, and tomorrow necessarily is going to be through my looking glass because I'm just guessing at what I think is going to happen next with seaweed. I think global warming is going to have huge impacts on the things we have in our area. And we're already starting to see some interesting patterns, and we'll see how they go. This Bonomosonia hemifera is very common to the south of here, southern Maine. You'll see all kinds of it. Very rarely found on the coast of Nova Scotia. I've seen no reports for the Bay of Fundy. 2009, 2010, we found it on Grand Banana. This thing's got these crazy hooks, and it tangles with everything. So this starts to take hold here. I'd be expecting I'll get a phone call in about 4 years from someone in aquaculture saying, I got this stuff growing all over my nets, and it's all tangled in them. I'm pretty sure we'll see that happen. And so this is a warm water thing that's making its way north. This is a cold water thing. This has been reported in the Bay of Fundy from the 1950s to the 1970s when people first started seriously looking for these sorts of things. I don't have a picture of it here. I've collected it in the Arctic, but I have not been able to find it in the Bay of Fundy at all in the last 20 years. It is simply not here. And so Maybe it's decided that it likes to move to the colder areas. So we are seeing these patterns, and it'll be interesting to see how these play out over the next 50, 100 years. The final story has to do with Churchill. I was asked to go up and work on the Churchill flora, and I thought, okay, what do we know about Churchill? So I went to the only actual book or anything that talked about the Churchill flora, which goes back to 1957. That shows you how little work's been done. Little known from the 3 or 4 papers which describe its flora. When they judge it, we have a relatively poor flora, maybe 50 species. What the heck do I want to go to Churchill for 50 species for? But there's belugas up there and polar bears, and so, you know, it's good diving. Go up and have some fun. So away we went, and we found a whole bunch of new species. 9 new species. We got all excited about these species. We got these new species in Churchill. The problem is we kept barcoding things from the Pacific, and sure enough, none of these were new species. They were all Pacific species. So we were finding all these new Pacific species in Churchill that weren't reported there previously. Now this is an interesting thing, because currently we believe that the Arctic flora is almost entirely of Atlantic origin. In fact, there are only 3 species that the Arctic and the Pacific share uniquely in common. And we've landed 9, and we're actually up to about 14 now, on top of these 3. This is kind of a weird pattern to be turning out. And so it got me thinking about how could this be happening? Is this again an introduction problem? Churchill's a grain port? Or somehow we're moving all these Pacific species into this port as part of these grain ships? Or are we starting to see the signs of global warming? Are we starting to see these algae moving their way across the Pacific towards the Atlantic? And I tripped across this really interesting paper. A biological consequence of reducing Arctic ice cover, the arrival of the Pacific diatom, Neodenticula seminae, in the North Atlantic for the first time in 800,000 years. Now obviously nobody was around to do that. They went out and they did sediment cores and they aged the cores, and they figured out the last time that species was in the North Atlantic. And in the year that they found this, there was a whole bunch of oceanographic data around that time that showed that we had increased flows of Pacific water into the Northwest Atlantic the summer before that, and we had an extensive ice-free northern period the year before that. And they're pretty sure that this thing came across the top with this Pacific water. In fact, we know during earlier warming events that most of our Atlantic flora and fauna actually does have Pacific origin. That's where our stuff came from 3 million years ago. And so this is very interesting that this pattern may be repeating itself in the current time. Well, it's a great big black box. We need a lot more sampling. We have samples from Churchill and this one trip to the northern tip of Baffin Island, and there is nothing barcoded through this entire area here to get a feel for what's going on up there in the Arctic. And Arctic sovereignty, Canadian government, no chance, no money. In fact, this trip here was paid for by the American government. I'm still trying to figure that one out. So, where are we with this? I think the evidence is leaning towards climate change. I think we're going to see some dramatic changes. And we got our first taste of this when we started looking at this Saccharina again. What we showed with our genetic work is that Pacific Saccharina latissima and Atlantic Saccharina latissima are actually distinct genetic entities. They're 2 very distinct genetic populations. And what we found for the plants in Churchill is they're actually hybrids of these 2. And that tells us that we have Atlantic plants moving into the Arctic, but we've also had these Pacific plants come the whole way across into the Arctic as well. And what we're seeing is that genetic signature is moving this way. We're not getting Atlantic signatures in the Pacific, we're getting Pacific signatures in the Atlantic. And so we're actually seeing this with this kelp species. We're seeing this movement from the Pacific to the Atlantic, and I think we're in for a real treat when this keeps going. I would predict that our new Arctic endemics, as we call them, are Pacific species caught en route to the Atlantic. I think that global warming is going to radically alter the benthic algal diversity of the Arctic and the Atlantic as more Pacific species join what's now called the Trans-Arctic Biotic Exchange. And I was so proud of this idea, and I ran down the hall to our population geneticist, and he said, "Gary, go read this paper." Coming Arctic Invasion. It's already been published. My idea got down the hall. This paper got into Science or Nature. I don't even remember anymore. It's so bitter. And that's how science works. So I think this is going to be an interesting time in biodiversity science in our area. So just to sum up, I think DNA barcoding is an amazing tool. It holds great promise for resolving a lot of the cryptic species issues we're dealing with, and it allows us to look at distributional limits of species, which is important. Big message, there are far more species on the planet than we currently realize, and that's exciting as a taxonomist to go out and find these things and name them. I think the levels of introduced seaweeds are far beyond what we currently realize, and this is going to become exacerbated by global warming. And that's something we're going to have to deal with. And unfortunately, people are like, what can we do about it? Absolutely nothing. There's nothing you can do. These things get into an area, you will not be able to eradicate them. You will learn to live with them. The ecosystem will learn to live with them. Or we stop all shipping on the planet. That might solve the problem in the short term. I think global changes in climate are really going to have impacts that are going to be rapid and substantial. And this diversity distribution issue will become a very exciting and hot topic of research. And I think the important issue is that the time to collect data is now. Species are going extinct at an unprecedented rate. Species are being introduced around the planet at an unprecedented rate. And species are changing their ranges at an accelerating rate. And once again, this isn't gloom and doom. I know it sounds like it maybe from an outsider, but this is exciting for us. This is the kind of stuff we study, and we're getting to see it happen. And so from the perspective of being a scientist who works on biodiversity, these are very interesting times. There are some neat changes occurring on the planet. So, I have to acknowledge my funders. Genome Canada has paid for a lot of the molecular work that's been done. NSERC, a lot of the molecular work that's been done. The New Brunswick Innovation Council, or New Brunswick Innovation Foundation and the Canada Foundation for Innovation have bought all types of molecular equipment for our laboratories. And I'll give them a little credit. They also bought us some scuba gear and things like that, so that was good. A lot of colleagues around the world are sending us samples. Tanya runs my lab and literally does all of the molecular work with the help of the students and the postdoc. And then I have this group that we call the FICA Ward. These are my students, my postdocs, and they're a wonderful group of people. They are the ones who do most of the work that I get to talk about, and they really give me a reason to be very, very proud for what I do. Thank you very much for sharing in my story.
Questions?
I don't know if you can answer the question, but how do they reproduce?
How do seaweeds reproduce?
Yeah.
Oh boy. Would you like to know green algae, red algae, or brown algae?
You've got time. The edited version.
Seaweeds have extremely complicated life histories. I couldn't even begin, even when I teach a fourth-year course, we just scrape on the surface. But they do have sex. There are boys and girls, and they are also, a lot of them are both sexual and asexual, so they can do clonal reproduction as well. But to actually get into the sexual patterns, they're extremely complicated. Yeah, there's some crazy things that happen. It's actually very interesting. What got me into seaweed in the first place was red algal life histories. When I first started learning about red algal life histories and all the crazy things that these algae can do, I was like, wow, that was it. So, it is very intriguing, but it's very complicated.
Why is seaweed a weed?
Seaweed is not a weed. Yeah, I don't know where the terminology comes from.
I can venture a guess for any boaters in here, it's because all the boaters trying to prevent seaweed from growing on the bottom of their boats and slowing them down.
That could be a reason, yeah. It's definitely a problem. In fact, one of the big funders of algal research is the U.S. Navy, for exactly that reason. They're trying to figure out how to keep the algae from growing on their submarines and their boats, because it slows them down and it makes them noisy. That's the other problem they have. So they spend a lot of money that way.